Hi everyone and welcome back to my channel. Today we'll be discussing about the CNS physiology, and in that today we'll be seeing the introduction part and about the glial cells, that is the supporting cells, and the blood-brain barrier. Okay, so in CNS there are mainly two types of cells. First of all, the first one is the neurons. The next one are the glial cells, that is the supporting cells. Neurons are like doctors in a hospital, and the glial cells are like the nurses. Neurons are about 10 billion in number, whereas the glial cells are 10 times more. Okay, you see, in a hospital usually the number of doctors are less, but these supportive staffs, the nurses, the cleaning technicians, engineers, pharmacists, they are more in number, right? It's like that. And neurons, they do not form after birth. Once they are formed before birth, the number isn't changed, whereas the glial cells can be formed during the lifetime also. In this, there is a pathogenic condition where the glial cells become neoplastic, thus forming the gliomas. Okay, so what are the functions of these cells? The neuron's function is simply to make electrical impulses and to send it or communicate it via the synapses. Synapses is the junction between two neurons. Actually, the junction between two neurons, that is known as a synapse. And the glial cells, so glial cells are in a variety of types. The first one is the microglia. See, the microglia causes phagocytosis, that is one of an immune response. Okay, phagocytosis is like eating up, I would say. And then there are the oligodendrocytes, which make the myelin sheath around a neuron. It's like, see, these neurons are there, right? Neurons are there, right? For example, this is a neuron. It is like an open electrical cable. So in an open cable, when electricity passes, it's not safe, right? It has to be insulated, right? It has to be insulated around. So this insulation is done by the myelin sheath. Okay, and this insulation of the axons in the CNS is done by the oligodendrocytes, and the peripheral nervous system is done by the Schwann cells. Okay, these oligodendrocytes can actually make one oligodendrocyte can myelinate approximately 30 neurons. And then there are the ependymal cells. These line the ventricles. Ventricles, not the heart ventricles, but the spaces between the brainstem and the brain. So over there, the ependymal cells are there, which contain CSF. And then there are the astrocytes. These are the main supporting cells. And what is its function? It basically supports the blood-brain barrier. See, this blood-brain barrier is made up of a few types, few components, I would say. So the components are the tight junction. You see the tight junction, the basement membrane, the astrocytes foot process, and the pericytes. So this is an pictorial representation. This astrocytes, which supports the blood-brain barrier. The tight junctions, it has claudin and occludin proteins, and these blood-brain barriers are supposedly it should actually separate the blood from the CSF. And these astrocytes help in maintaining the CSF composition by removing the metabolic waste, especially the potassium. Okay, it's like these metabolism occurs even in the brain, and certain waste materials are discharged out. So these have to be removed, right? It is done by the astrocytes. And also, it also makes nerve growth factors. You see, new nerves can't actually be formed, but the nerve can actually be repaired and regenerated. And during fetus development, right, these glial cells or these astrocytes show the direction to the neuron and place the neurons in the right position. So yeah, the blood-brain barrier is made by tight junctions between the endothelial cells. These endothelium means the blood vessel is there, right? These blood vessels have an epithelium, and that epithelium is known as an endothelium, which is made up of tight junctions. And this blood-brain barrier prevents substances to pass from the CSF to the blood. And only lipid-soluble substances can cross, whereas water-soluble, see these lipid-soluble substances, right? It crosses via diffusion. Okay. And then there are these, I would say, glucose and amino acids are there, right? They go via facultative transport mechanism. Okay. So yeah. Okay. Now what is the CSF? That is the cerebrospinal fluid, and performs a lot of similar functions as of the blood, I would say. Not specifically blood, more specifically I would say the plasma. Okay. It maintains internal environment for the neurons, which are the pH level, the temperature, and the osmolarity, and provides nutrition and removes the waste also. And the CSF acts as a shock absorber and prevents from any vast injury. The CSF's composition is similar to plasma, but you see, the CSF composition is similar to plasma, but lesser in quantity, I would say. And for example, you see the protein levels and glucose levels are considerably low in the CSF when compared with the plasma. Certain exceptions are there where it is more in the CSF, that is the chloride ions, bicarbonate ions, partial pressure of CO2, and magnesium ions. Okay. There is a condition called meningitis, which is caused by bacteria and viral infections. Okay. And this CSF findings can help in distinguishing it between a viral meningitis and a bacterial meningitis. For example, if it's a bacterial meningitis, what happens? There is the presence of a bacteria. There is a presence of bacteria, right? Bacteria require energy to grow. Therefore, it consumes the glucose and decreases the glucose levels. Whereas in a viral meningitis, the glucose level is viruses don't require actually glucose, so it's somewhat little less or in the normal range itself. And during inflammation, the protein levels in the CSF increase. Where are these CSF actually present? It is present in the ventricles and subarachnoid spaces. And the CSF is secreted by the choroid plexus and is absorbed by the arachnoid villi. So the normal volume is about 150 ml. Okay. And the pressure in it is 5 to 10 mm of Hg or 50 to 130 mm of water. So the daily production and absorption of CSF is 500 ml per day or 20 ml per hour. You see, when there's a lot of CSF produced, there is rise in intracranial tension, which leads to Cushing's reflex. This Cushing reflex occurs during head injuries, and it causes reflex bradycardia. That is, there is an increase in blood pressure, but there's a decrease in heart rate. So for the treatment, 10% of mannitol is given to maintain it. So this is about the introduction part, the glial cells and the blood-brain barrier in CNS Physiology. And I hope you liked this video and you gained more knowledge through it. Thanks for watching, and if there's any doubt or you want to let me know anything, type it down in the comment section and share it with your friends. And if you haven't subscribed yet, please do. And as always, be awesome.